Smooth muscle cells surround hollow structures like blood vessels, and when they contract, the lumen gets smaller, which affects blood flow. So here, we'll take a more in-depth look at smooth muscle characteristics and contraction. So smooth muscle cells are these spindle-shaped cells that are organized in sheets and surround hollow structures and organs in our body. So these cells do have actin and myosin, but they don't have the striations that we see in cardiac muscle. And that's because in smooth muscle, these thick and thin filaments are organized diagonally. They're not organized into sarcomeres. During contraction, the thick and thin filaments interact and form cross bridges, and they slide past each other. And when that happens, it causes the cell to balloon out, like we see here in this contracted image. Smooth muscle cells have a very well-developed sarcoplasmic reticulum, so it makes sense that calcium is a big part of regulating contraction, and the amount of tension developed upon contraction of smooth muscle cells is proportional to the intracellular calcium concentration. Contraction may be in response to an action potential or may also be in response to a subthreshold depolarization where no action potential is fired. So there are two different types of smooth muscle, multi-unit smooth muscle here on the left and unitary or single unit smooth muscle on the right. And you may already be able to tell just from the images, but the main things that separate these two types of smooth muscle are the amount of innervation and the number of contacts between the cells. So multi-unit smooth muscle cells are densely innervated, but there are few gap junctions to connect the smooth muscle cells. So this type of electrical isolation really allows each smooth muscle cell to contract independently of its neighbor, and this allows a really fine motor control. And this type of smooth muscle is found in the iris of the eye, for example, where you want that really fine control. And then in this single unit or unitary smooth muscle, there's much less innervation, but much more electrical coupling between cells through these gap junctions. So there's lots of gap junctions between these smooth muscle cells. And that allows a group of cells to contract together in a coordinated way. So the bladder, for example, is surrounded by single unit smooth muscle so that contraction and emptying of the entire bladder can happen all at once. In reality, most smooth muscle in the body falls somewhere on a continuum between these two types of smooth muscle, and that includes vascular smooth muscle, which will be a major focus of the cardiovascular system. So smooth muscle contraction is initiated by an increase in intracellular calcium. And that calcium is important in regulating a couple of key enzymes that control cross-bridge binding and cross-bridge release. So that calcium can come from a couple of different sources. It can come from the extracellular fluid through calcium channels that may open based on a couple of different mechanisms that we'll look at in a minute, or it can be released from the sarcoplasmic reticulum. So once that intracellular calcium rises, it triggers cross-bridge cycling and contraction through this variety of steps. So first, calcium binds to this protein in the cytoplasm, calmodulin, and that calcium calmodulin complex binds to and activates the myosin light chain kinase. And when activated, the myosin light chain kinase phosphorylates the myosin light chain, and that phosphorylation changes the conformation of the myosin head, allowing myosin to bind actin, initiating cross-bridge cycling and producing this tension or contraction. And it's important to note that the amount of tension generated by a smooth muscle cell during contraction is based on the amount of intracellular calcium. So with higher intracellular calcium concentration, there are more cross bridges bound, and therefore more tension can develop with the cross-bridge cycling. Myosin light chain phosphatase dephosphorylates that myosin light chain, causing cross bridges to uncouple and leads to relaxation. So this myosin light chain phosphatase is constitutively active, so it's always active. So it's really the relative activity of each of these enzymes, the myosin light chain kinase and the myosin light chain phosphatase, that dictates the contraction versus relaxation of the cell. So as we just saw, smooth muscle contraction is stimulated by this rise in intracellular calcium concentration. And there are multiple mechanisms that are involved in calcium entry into a smooth muscle cell. So voltage-gated calcium channels are one. And voltage-gated calcium channels are on the plasma membrane of the smooth muscle cell. And they open when there's depolarization of the cell membrane, which then allows the calcium to flow down its electrochemical gradient and into the cell. And this depolarization that opens the calcium channels can be either subthreshold depolarization, where an action potential is not fired, or it can reach threshold and cause an action potential. So either of those cases would open voltage-gated calcium channels and allow calcium influx. There are also ligand-gated calcium channels on smooth muscle cells, and these aren't regulated by changes in membrane potential, but instead they're regulated by various hormones and neurotransmitters that bind to their specific receptors, which are coupled by a GTP binding protein to these calcium channels. So when the hormone or neurotransmitter is bound, the ligand-gated calcium channel is open and calcium can flow into the cell. There are also these IP3-gated calcium channels that allow calcium release from the sarcoplasmic reticulum. So to activate this calcium release from the sarcoplasmic reticulum, it happens here on the plasma membrane of the smooth muscle cell. So hormones and neurotransmitters act with their specific receptors, and these receptors are coupled by a G protein to phospholipase C. That phospholipase C hydrolyzes this phosphatidylinositol 4,5-diphosphate, or this PIP2, to IP3 plus diacylglycerol. And it's this IP3 that diffuses to the sarcoplasmic reticulum, where it opens calcium channels, which then releases stored calcium into the intracellular fluid. So this is a few different mechanisms that are able to increase the calcium concentration inside the smooth muscle cell and initiate that contraction.